I just want to revisit this topic we covered a little while ago. We talked about basically what a species is. And now with this maybe deeper perspective on morphometrics, we can look at these species boundaries with a more critical eye and say, we don't always understand exactly where the boundaries are among species, and being able to identify them using morphology can be difficult. So my lead-in image for this topic is a graph from a paper that was studying a species called Arabidopsis lyrata. And these areas with red here, these are kind of on the fringe, this boundary between different populations that have different reproductive strategies. So selfing populations and different genetics outside of these boundaries. So potentially a name worth applying to these different groups of the same species. Same thing over in the United Kingdom. This is a population of, or a set of populations of these plants in the genus Geum. Rather different plants, right? Different flower form. We got a pendant flower here and one with more open flower with different colors and so on. But in this range where the two species are overlapping, there are some mixed populations and potentially some opportunities for genetic exchange between those different populations of different species. So being different, this can be an area where the morphological differences are important for keeping the genetic differences between these species. This was something that I worked on. I think I showed you graphs like this previously as well. Having a couple of species, these are both native to Eastern North America, and the features that tell them apart were broken down into these quantitative measurements. And you can basically determine based on a combination of measurements which one of these features or combinations of these features would be able to tell you if you have one species or the other. We ended up with, I think, 95% of the samples were able to be identified using these morphological features. So sometimes it takes some investigating. So once you've identified your variation, using that variation among the morphological features to break your, basically decide where one species ends and another one begins, you need to account for some of that variation. So here we've got range of values. This is a diameter of stem, and this brown species has 90% of its measurements greater than this. And looking at this 1.2 millimeter cutoff, you can also take that as a cutoff where it's different from the blue species. So if you break the blue species from the brown species at 1.2 millimeters, just with this one measurement, you're accounting for 90% of the blue species and 90% of the brown species. Now, it doesn't count for everybody. And so you also might want to tell people about this extended range of variation where not many samples fall, but enough fall to make it important to make that distinction, that some of the brown species can exist down here all the way down to 1/2 millimeter, and some of this blue species can exist all the way up to 2 millimeters. And this, of course, is just one measurement for a species that have lots of qualitative and quantitative features to use. So combining lots of different traits allows you to really make that distinction between species and potentially diagnose all individuals of the species. So just a little highlight here. This is how these measurements turned out into the key. I've got the parts highlighted where these inflorescence stems are the measurement. So we've got these 1.2 to 5 millimeters, 0.4 to 1.2 millimeters. So kind of conveying that there's some confidence in having that 1.2 millimeter cutoff, but also this range that extends it to a bigger area in the blue species and smaller measurements in the brown species. And you can do this across a variety of species. This is a study that I did on a whole bunch of different nymphoid species in Southeast Asia. And again, with one measurement at a time, sometimes you can make a pretty clean distinction between one species and another. Sometimes you've got considerable overlap. So if you have this blue species against this green species, they're mostly different from each other in the size of their calyx lobe. But a good number of individuals would have overlapping ranges here. Same thing for these other measurements. So you can have very different means. The blue mean here is a lot different from the orange mean, but lots and lots of overlap. So like a meaningful difference, but not necessarily one that you could use to diagnose these two species away from each other. One way to combine all these different measurements together, kind of in an investigation to determine whether you actually have different species, you can do what's called here this principal components analysis, which is a statistical method where you take lots of quantitative measurements and you just mush them all together into one big analysis. And it kind of squishes your data down its many dimensions, and it squishes it into 2 dimensions. And you get the biggest differences on the x-axis here. And you can see the brown, mostly brown, is over to the left side and mostly blue is over to the right side. Not a whole lot of overlap. You can see there's kind of a split there. So this is telling us kind of mathematically that there's enough difference that if you combine all these different measurements, you can reliably tell most of the browns from most of the blues. And then it tells you what the different features are that are most useful for making that distinction. So calyx length, blade length, and so on. And then these ones on the y-axis are kind of a secondary differentiating factor: pedicel length, stem diameter. And this technology is moving forward. So we're getting into this territory where in some cases you can like set a computer and then walk away. This takes advantage of artificial intelligence, statistical methods, computer programming. And if you have, like they have here, these herbarium specimens, you could teach a computer to draw shapes and say, okay, I need you to outline the leaflets on this compound leaf and determine the measurements, the dimensions of the terminal leaflet. And potentially you can do this with something you've already got scanned without having a person involved who gets in there with a ruler to actually measure anything. So pretty neat prospect for the future, and just keeps getting more interesting, more complicated from there. So being able to identify not only a couple of related species, but this effort to identify any species is getting a lot of steam. So people have a lot of hope for artificial intelligence and the ability of computers to train on images and in some cases learn better than people might be able to do. I'd say probably you can't beat the experience of an expert, but if you're a person coming at these leaves for the first time, you would probably benefit a lot from having a computer help you along, being able to rule out possibilities that are possible or not possible, and so on. So this is just a little schematic about identifying different woody plant species. Actually, not all woody species, but distinctive leaf shapes, and then kind of grouped by their similarities. And this is the basis for a few different apps. You can get apps that will get them on your phone. You take a picture of a plant, and it uses this machine learning artificial intelligence to tell you what it thinks you've got. So kind of rules out possibilities that are not possible. And because it's machine learning, it can take all kinds of factors into account. So color, hairiness, angles, shapes, all kinds of interesting information. And some of these take into account geographic information. They'll tell you plants that are more likely to be ones you're running into based on where you're located. And there are lots of apps like this. I just grabbed a few screenshots from some of these maybe more prominent ones. Ever changing. Some of the ones that are more popular are ones you need to pay for. But if this is something you want to use in the field, this is a good way to, I'd say, get started on maybe getting closer to having the right identification off the bat. Still, I'd say not a 100% substitute for knowing your plants, for being able to identify them yourself without using an app. But these days, I can't really deny that this is a reality. This is a big part of our current reality, is that this is kind of an option for people. And why not use the tools available to you, right?